We're going to speak about the relationship between pre-implantation morphokinetics of human embryos and sex chromosome pattern. Embryonic gender identification techniques were born out to help the people to choose the gender of their offspring. Sex selection techniques are using for two main purposes. Medical purposes to avoid an gestation of fetus carrying X-linked genetic anomalies. Second purpose is to including social, yani for family balancing between male and female. Ultrasound technology can be used to identify the gender of fetus, of course, but not before the first trimester, as you know. Nowadays, it's possible to know the sex of embryo at the very early stage of embryonal development before transferring it to the uterus, especially for those patients undergoing in vitro fertilization treatment. Actually, we use two techniques for investigating the karyotype or genetic pattern for the embryos, PCR or fluorescence in situ hybridization, just to know the numerical anomalies and also to detect the sex of embryo. We can see in this slide two type of karyotypes, one for XY and 46XY in the left side and 46XX for female embryo. Recently, researchers have tried to understand the relationship between early embryo morphokinetics and the sex of the embryo. We mean here with the early morphokinetics, the embryo diffusion from day 0 after egg injection until day 5 before transfer the embryo to the uterus. It was suggested that during pre-implantation development, the embryo may show different morphologically features according to its sex pattern. These variations in the embryo's phenotype can be attributed to various in the sex chromosome component. Could you please click start the video here, Dr. Faiza?
Yes, can you repeat what you want me to do?
Yeah, there is video on this slide. Could you please just press the video? Show the video to see to show the embryo morphokinetics from day 0 until day 5. This video taken by time-lapse microscopy, special incubators provided with the special camera to monitor the embryo diffusion from day 0 until day 5 before transferring the embryo to the uterus. The idea, we try to understand if the X chromosome has any effect for the behavior of embryo diffusion within this period or not. Yeah, this is the blastocyst stage before transferring the embryo to the uterus. Time-lapse was recently used to determine if there are any relationship between embryo sex and early diffusion behavior of human embryos and animal to help the in sex selection. Please go for next. After next slide, Doctor.
Okay. Is it or
next, please? Next.
Okay.
This study was aimed to determine the relationship between the embryo gender and its chronological changes during the two pronuclear stage, also embryo diffusion stage, as well as blastulation time on day 5. We used the time-lapse imaging and BGT techniques.
Should I move next?
Yes, okay.
Okay.
This study was conducted at the IVF Center Rayyan in Saudi Arabia. 120 IVF cycle with BGT were included in the study. Depending on the BGT result, only the normal embryos were included in this study. 416 embryos divided to two groups, male including 227 embryos and female 189. In the result of this study, the result revealed that there was a statistically significant differences comparing morphokinetics between male and female embryos. Firstly, we found there is difference between male and female in the tubian appearing, which in the slide showing that the XX embryo, the tubian and the XX embryo appear sooner than or faster than the XY embryos. The time for zygote formation in the XX embryos occur within 9 hours, whereas the XY embryos occur within 9.5 hours. Next stage is 2BN feeding. The 2BN feeding was faster in male than female. It takes around 22 hours in the male embryos, whereas the female embryos taking 24 hours. Last stage is the blastulation rate. We noticed that the blastulation rate in the female and between the female or XX embryos was higher than in between the XY embryos. Could you please go for next slide, Dr. Faiza? No, next slide. Here, okay. Can you show the video, please? In this video, we can see comparison between two different embryos. The left side, the one in the left side is the pink one is the female, and the green one is the male. You can see the difference between the. Can you go from the beginning, Doctora?
Okay.
Yeah, represent the video. You can see, especially in the 2BN feeding, we can notice the male embryo was faster than the feeding of 2BN happened earlier in the XY embryos. Yeah. Also, we can notice in the blastulation or blastocyst stage, the XX embryo, the blastocyst in the XX embryo was faster than in the XY embryo. Next slide, we can go for discussion.
Yeah.
However, this issue is still controversial. The detection of the earlier developmental differences between male and female embryos in vitro requires ideal condition in the form of high glucose and amino acid media. We mean in the culture media. The pattern of sexual genetics is responsible for the differences between male and female as reported by some researchers. Also, 6 hormones contribute strongly in the sexual differences of adult tissue. Okay, in the earlier stage of embryo division, 6 chromosome pattern reflects some of the apparent contrast between male and female, resulting in different in their proteomic and metabolism process. As reported by some researchers, in bovine, X-linked genes may be responsible for embryo morphokinetics differences between male and female embryos. Moreover, the faster rate at preimplantation may be attributed to messenger RNA expression of two genes, hypoxanthine, phosphorybosyl transferase, and glucose 6-phosphate dehydrogenase genes. These 2 genes are located in the X chromosome, and we can understand from this interpretation, no, the X chromosome, which is higher in female, has more chance to be faster than the male embryos, which has only one copy of X chromosome. Other studies showed that embryo differ in vitro in glucose and amino acid metabolism according to the gender. And it's mean that the 6 chromosome play important role for behavior of embryo division in the earlier stage. In the conclusion, according to this study, we conclude that the X and Y pattern show different fertilization. The X and Y sperm show different fertilization behavior. The rate of appearance and feeding of the 2B in stage, as well as blast rate at day 5, is influenced by the gender of embryo. We postulate that this may be used to help determining the sex of embryo within 1 to 2 until day 5 of embryo division after XC, without costing the for genetic study. And we can decrease the cost of the genetic test, and we use the embryoscope, the time-lapse embryoscopy incubator, to help for determine the X and Y, the XX and XY embryos. Also, we need more study in this field, as we see it's very important to explain more about this phenomenon. Thank you so much.